I'm Dr. Mary Mendonça. I'm a professor in the Department of Biological Sciences here at Auburn, and I've been here about 25 years or so, and the lab has been doing research on various things, but it really centers on um, how the environment causes differences in stress responses and different behaviors associated with reproduction and coping with stress.
Stressor is an environmental challenge or stimulus that causes a change in an animal's physiology. Stressors can include an extreme change in temperature, lack of food, and being hunted by a predator. Stress physiology is the study of the way the body responds to these stressors. Responses can be short-term or long-term. Short-term stresses are adaptive and can help the animal, while long-term or chronic stresses can cause the animal harm or negatively affect their health. Estrogen is a sex hormone found in animals that is essential for physical changes, development, and reproduction. It is found naturally in both males and females of all vertebrate species and is an important element of natural development. However, today it is being found in alarmingly high amounts in animals' food. This is the American alligator, found in the freshwater rivers, lakes, and swamps of the United States. It is an apex predator, sitting at the top of an illustrious food chain. Whatever its prey eats, it eats as well. Unfortunately for the alligators, they have found an unexpected threat. Estrogenic compounds in the form of herbicides and pesticides are leaking their way into the alligator's environment. The estrogenic compounds we use at our convenience have an unexpected cost. They mimic the sex hormones of these alligators. These compounds can influence development and reproduction in reptiles and could affect things like gender, reproduction, and perhaps even the immune system. This is Caitlin Murphy, a graduate student working in the Mendonça lab. Caitlin's research hopes to unearth the effects of these estrogenic compounds on the alligators and shine a light on the shadowed effects of our pesticides and herbicides on the ecosystem. Caitlin knows that the bacteria found in the guts of the alligators plays a central role in processing estrogen. She hopes to examine the difference in the gut microbiomes of normal alligators and those of alligators exposed to extreme levels of estrogen to further explain the effects of estrogenic compounds on the American alligator. The gopher tortoise is the only tortoise found in the southeastern United States. While unimpressive to the naked eye, they play a surprisingly powerful role in their ecosystem. Their distinct burrows offer shade from the southern heat and protection from predators. Unfortunately, the gopher tortoise is a victim of deforestation, poaching, and vehicle-related killings, causing it to be a threatened species. And now, gopher tortoises face a threat their burrows cannot protect them from: a deadly disease of the upper respiratory tract caused by a bacterium called Mycoplasma agassizi. One result of the disease is an acute infection that strikes hard and strikes fast, resulting in almost immediate death for the tortoise. Another result of the disease is a long-term chronic infection that destroys the tortoise's nasal epithelium, erasing one of the tortoise's most important senses, smell. Megan Kelly studies the effects of Mycoplasma agassizi in gopher tortoises with the hope of treating them and giving them the status and protective benefits of an endangered species. One aspect of particular interest to Megan is the disease's effect on reproduction. Smell is an essential aspect of communication for gopher tortoises. Male tortoises have large glands underneath their chins called mental glands that produce a mucousy secretion and a strong smell. The size of the tortoise's mental glands and strength of the smell is essential in establishing dominance and choosing a mate. Without this sense, the tortoises are helpless. Here is an infected tortoise placed in a pen with two model tortoises, one with a smear of mental gland secretion and another with nothing at all. Normally, the tortoise's sense of smell would allow it to distinguish between the two. But the infected tortoise is all but helpless. It can distinguish between neither tortoise and barely seems to acknowledge either one. Instead, it paces agitatedly around its pen, looking for a way out. On the other hand, healthy tortoises' mating behaviors are as interesting as they are belligerent. In an attempt to initiate mating, the male tortoise tries an extravagant technique, head bobbing. His attempt goes largely unnoticed, but nonetheless he attempts to mate with the unconsenting female. Unfortunately, despite his efforts, this tortoise's love is unrequited. This is the cane toad, found in areas of central and south Florida. When threatened, the toad secretes a highly toxic substance from large glands located behind its neck and along its back. However, its home is not Florida at all. The cane toad is actually native to areas of Central and South America, where it thrives in the hot, tropical climate. Its predators are largely aware of the poisonous danger they pose and for the most part avoid them. But in the 1930s, they were brought to the United States as a form of pest control, and they have since reproduced prolifically. Here, they are unwelcome invaders in a hostile land. Now they are moving north and are experiencing much higher levels of stress here than in their natural habitat. Stephen Gardner works in the Mendonça lab, studying the stressors that affect cane toads in the United States and how these stressors affect their behavior and immune response. When threatened, the toad's endocrine system mounts a response. The toad experiences a surge in adrenaline and hormones called glucocorticoids that are essential in the fight or flight response. If the toad needs to react to an immediate threat, this response serves the toad perfectly. However, here, in the United States, the chronic stress the toads have experienced have led to elevated levels of these hormones throughout their bodies. Over time, this has an adverse effect on the toad's health and behavior. To simulate a threat, Stephen prepares an adrenaline solution with which to inject the toads. After weighing the toad, he injects them with 1 micromole of epinephrine per gram of body mass. The result is similar to that of a live threat. Droplets of the waxy poison emerge from glands on its neck and back. Normally, cane toads in their natural environments are less targeted by predators and will move freely and are less prone to secreting poison when threatened. But the invasive toads, in a new environment with new threats, must change their behavior to adapt to the new stressors. To test the differences in the cane toad's reactions, Stephen constructs a closed track, inside which he plans to stress the toad by poking it. Stephen's test has revealed an interesting difference in the invasive cane toad's response. Cane toads on the invasive front will not hop around to avoid a threat. Instead, they remain still and secrete poison, a defense mechanism that will cause the attacker to come into contact with it and suffer the consequences. Stephen's research hopes to further explain how elevated stress levels affect cane toads' behavior and affect their immune response.
They'll get a very solid background in physiology, physiological ecology, endocrinology, how hormones mediate so much of our physiology and our behavior.